T cell activation and the major histocompatibility complex, MHC. T cells are white blood cells of the lymphocyte variety, and they are key players in your acquired or adaptive immune response. They come in two varieties, helper T cells and cytotoxic or killer T cells. The helper T cells are important immune system coordinators and are responsible for activating both B cell lymphocytes, which make antibodies, as well as the cytotoxic T cells, which kill altered, infected, or even foreign human cells introduced into your body, as would be the case in an organ transplant. T cells are activated to respond when the T cell receptor on their surface recognizes an epitope of an antigen being presented by the MHC molecule on another cell. Again, there are two varieties of MHC molecules, which correspond to the activation of the different T cell varieties. MHC1 interacts with cytotoxic T cells, and MHC2 interacts with helper T cells. Notice this figure showing the interaction. You can see that for each scenario, there is one additional important molecule. Let's go over this. For a cytotoxic T cell, the T cell receptor binds to a foreign molecule presented by MHC1, and this interaction is stabilized by the CD8 molecule on the cytotoxic T cell. Remember that MHC1 molecules are present on all nucleated cells of the body. The helper T cell receptor interacts with an epitope presented by MHC2 molecules, and this interaction is stabilized by the CD4 molecule on the helper T cell. MHC2 molecules are only present on the professional antigen-presenting cells of the body, such as macrophages, dendritic cells, and B cells. How does an epitope of an antigen end up in the MHC molecule? First, we must clarify that antigens in the body can be found in one of two locations. If the antigen is outside of a cell, it is termed exogenous. Bacteria and debris are exogenous. Antigen may also be presented by the cell itself. Cells may produce normal components, or it may produce antigens if it is infected or altered in some way. Virally infected cells would produce endogenous antigens. This next figure clarifies how these different antigens end up in the MHC molecules. For an antigen produced within the cell routinely will sample its own contents and display epitopes with MHC1. This is termed the endogenous pathway. All cells do this. It is the way the body can police its cells and ensure that they are functioning properly, meaning they are not malfunctioning as a cancer cell might, nor are they infected. A cell infected internally with a virus or a bacterium, or if it was mutated, would display abnormal epitopes on the surface, which could be recognized by cytotoxic T cells. For an antigen that was external to the cell, it could be internalized by phagocytosis in the case of a phagocytic cell, or endocytosis as in the case of a B cell, whose B cell receptor latched onto an antigen. These internalized antigens are digested and coupled with MHC2 molecules and presented on the surface. Hence, this pathway is referred to exogenous processing pathway. This next figure compares and reviews pathways and the interaction of the T cells with MHC. On the left is shown the endogenous pathway and the interactions with the cytotoxic T cell with the epitope presented by the MHC1. On the right is the exogenous pathway with the helper T cell interacting with MHC2. If a helper T cell interacts with an epitope of MHC, it is stimulated to proliferate, termed clonal expansion. Some of the cells will be active and secrete cytokines to activate either B cells or cytotoxic T cells. This depends on the type of cytokines released. T helper type 1 will secrete cytokines to stimulate cytotoxic T cells to action, while T helper type 2 secrete cytokines to activate B cells to secrete antibodies. Other helper cells will be memory cells for a future response to this epitope. When a cytotoxic T cell interacts with an epitope of the MHC, it will wait for the cytokine stimulation of a helper type 1. When received, it too will proliferate. Some cells will then be activated to secrete perforin and granzyme to kill the altered cell. Others will become memory cells, an arsenal for future responses. This concludes my brief review of MHC and T cell activation.